Hi everybody, and welcome to the Micro E-Learning Program from the Global Parkinson's Genetics Program and the Aligning Science Across Parkinson's. My name is Mauricio Chaparro. I'm a medical geneticist and currently a molecular medicine PhD candidate at the Cleveland Clinic. Today, I will talk about genetic variant prioritization and classification. In this video, we will briefly introduce variant calling, as well as genetic variants classification and prioritization according to the American College of Medical Genetics and Genomics guidelines. Let's begin. Regardless of the method used to obtain genetic information from your patient or research participant, whether it is genotyping arrays or next-generation sequencing, there's a point where both methodologies converge. This common point is variant calling, where the genetic variants are identified by comparing the obtained results to a reference genome. If you want to learn more about file formats, check Module 5 from the Beginner Bioinformatics for PD Genetics course. After completing the variant calling, we proceed with variant prioritization and classification. But what does this mean? Let's start with variant prioritization. On average, a person can carry over 5 million genetic variants. But which of these numerous variants are relevant to my research or even to my patient? We are primarily interested in finding genetic variants related to specific diseases, such as Parkinson's disease. However, with thousands of variants to sift through, we need to apply filters to prioritize only those potentially related to Parkinson's disease in this case. When filtering for variants causing or contributing to disease, we typically think about variants that are located in genes biologically related to the phenotype of interest, have a very low allele frequency in the general population, usually below 5%, or in other words, are rare variants, change the structure or function of the resulting protein, such as nonsense, missense, French shift, and those other located in the splicing sites, among others, are predicted to be damaging or deleterious according to in silico predictors or functional studies, are variants previously reported to be associated or within a gene where other variants have been previously linked to our disease of interest? And for disease-causing variants with high penetrance, it is imperative that they segregate with disease in more than one family. We retrieve all this information for each genetic variant using automated algorithms that search multiple databases for relevant data. Some examples of these databases are: for allele frequency and type of variant, GNOME-AD, which includes exact data, and DBSNP. For in silico prediction of variant effect, mutation taster, SIF, polyphen, and CAT. And for previous associations between a variant and a disease or phenotype, ClinVar and the Human Gene Mutation Database are examples. This complete procedure of assigning each variant specific details is known as variant annotation. During this process, we may find that some prioritized variants have already been reported as pathogenic in public databases, or in silico predictors may classify them as disease-causing. Are we done with this? No, the job isn't finished yet. Even though the variant annotation process is completed, it only provides separate information about a genetic variant. Each piece of information needs to be interpreted as a whole. This will be the variant interpretation and classification process. To accomplish this, in 2015, the American College of Medical Genetics and Genomics released guidelines allowing the classification of variants in a standardized way into 5 different groups: benign, likely benign, variant of unknown significance or VUS, likely pathogenic, and pathogenic. For variants classified as pathogenic and benign, there is almost 100% certainty of their effect. For likely pathogenic and likely benign variants, there is a greater than 90% certainty of their effect, while for VUS, available evidence is inconclusive. The ACMG grading system relies on several criteria, many of which are similar to those used for variant annotation and prioritization. When a criterion is met, a point is assigned to either the pathogenic or benign side of the classification spectrum. Depending on the total number of points, variants are classified using the cutoffs presented in the guideline. Variants that do not surpass the cutoff for pathogenicity or benignity remain classified as VUS. Avoid making diagnosis or providing genetic counseling based on variants classified as VUS. Some final tips to consider. Please Note that none of the classifications are made with 100% certainty. As genomic sciences evolve, variant classifications may change over time. Additionally, ACMG criteria are not rigid, and clinicians or researchers can weight each criterion differently. Some criteria rely on information mainly available to the clinical or research group. While these guidelines recommend several databases and bioinformatic tools, it's essential to consider that many more options are available now compared to when they were initially published. Currently, there are online platforms that assign many of the ACMG criteria, supporting and accelerating the variant classification process. However, don't rely solely on them. As mentioned before, you may have valuable information not publicly available that supports variant classification. Lastly, remember that these guidelines are only for classifying rare variants associated with monogenic diseases. They should not be used for classifying common variants associated with complex diseases. In summary, variant calling and annotation are only the initial steps to variant interpretation or classification. Ensure that you are annotating your data using the most updated resources, using softwares like Annovar or VEP. Bioinformatics tools are supportive but do not solely determine the classification of variants. Use all available information to interpret your findings. BUS remain as such until proven otherwise. Monitor them over time until reclassification is achieved. And do not rely on research findings to inform the patient. Every single finding needs to be confirmed by a CLIA-certified laboratory. Thank you for listening, and I hope you enjoyed this video.